Hey, 4C divers! Thank you for tuning in tonight. We are super excited. We have a fantastic presentation for you guys. But first, you know what you need to do. You need to hit that like button, that thumbs up, that heart emoji, that smiley face. Let us know that you are here and that you're listening in. In the comments section, you can say hello. Tell us where you're listening in from. We always love to see you guys and have you guys participate in our comment section. And also, if you did not register for tonight's presentation, you're going to want to do that. Go to our website, www.force-e.com. Go to the event tab, scroll down, find tonight's presentation, register before 6:45, because guys, we are giving away something really awesome. We have never done this before. We are giving away a chance to go with a 4C dive guide to the Blue Heron Bridge to go and seek out these beautiful seahorses that are there right now, and a bunch of them are out there mating. And so we have been seeing photos on Facebook and Instagram lately. So if you want to go and see some seahorses underwater and you live close enough where you're going to be visiting this area, maybe you will win tonight. We'll do that raffle at the end, so make sure you register. If you don't register, we don't have your name to put you in our random name picker. So go to www.force-e.com. And while you're there on our website, it is travel month here at 4C. So guys, if you love to go and travel around the world and see new things and you want to do it underwater, do it with 4C. We've got lots and lots and lots of trips planned for you guys. We actually have one at the end of the month. It's just a day trip going over to the Bahamas to go play with the hammerhead sharks over in Bimini. We have a trip planned to Raja and Pot later on in the year in April. We have the Roatan trip, we have the Philippines trip, and we just added our Cayman Brac trip in December. So if you want more information, go to our website, click on our travel month banner. You can find out more about these trips. And guess what, guys? Seahorses live in these places too. So you can go and see the seahorses over all around the world if you want to do a tour of seahorses. Anyways, guys, thank you for tuning in. If you don't know my name, and some of you, it looks like some new people signing in on here. My name is Nicole. I'm your social media gal here at 4C. And I never had anybody do a presentation, whether it was in our store or on Facebook Live, about seahorses. And so I was trying for years and years and years to find someone that could help me with this. And I finally, insert Dr. Emily Rose. Hi, Emily.
Hi, I'm so excited to be here with you guys today.
Excellent. So, guys, I am super excited because I love seeing seahorses. I have a degree in marine biology, but I don't know much about these little critters. So hopefully you guys and I are going to learn some cool things today. And again, go to our website and register so that you can go and see the ones at the Blue Heron Bridge with one of our dive guides, because we're giving away a dive guide with a Blue Heron Bridge expert 4C instructor / dive master. So all right, so Emily, I'm going to go ahead. I'm going to knock off here, and the floor is yours.
Awesome. Thank you. Okay, so as Nicole mentioned, my name is Dr. Emily Rose, and I've been working on seahorses for approximately half of my life now. And I'm currently based out of Valdosta State University, but I'm going to give you a little bit of my background and kind of how I have come to working with seahorses. So when I describe my goals in life, it's typically wanting to be Miss Frizzle from the Magic School Bus. And so I wanted to kind of take science and join it with being a teacher. And so being a college professor is as close as I could get, basically. So I've spent most of my career training as both a scientist and also as a teacher. And turns out I've been working on these really cool creatures for my entire career. So cygnathids are the fish from seahorses to pipefish to sea dragons. And all of these fish have something in common, which is male pregnancy. So we study these creatures because they're found all over the world. You have seahorses found in a ton of different locations. So as Nicole mentioned, right, you can go down to Roatan and see them. You could go to Australia and see them. You could go to the Indo-Pacific and see them. And so these are going to be found all over. The sea dragons are only found Australia area, so you're not going to see any of those off the coast of Florida. But it's a really interesting group of fish because they all have pregnancy in the males, and they also are very, very cryptic animals. They're not very good swimmers. They have to hide for the most part. But how they mate is really interesting. You have seahorses, and the pipefish on the top are going to be monogamous fish that pair bond together. Then you also have species where the pipefish here in the middle that's underneath the seahorses with the red and blue stripes, they're going to be ones that are going to have lots and lots of mates throughout their breeding season. So there's a lot of differences in how these fish mate, which is what we find so interesting. So my background is studying the research program consists of how these fish have evolved over time. Why are some monogamous and why are some promiscuous with their mating processes, right? We also want to know more about their ecology. Are they found in a seagrass bed, a coral reef? Are they found on the coast? You know, what type of habitats do they live in? What do they eat, for example, right? And then mostly we're really interested in the environmental component because humans are constantly altering their habitats and introducing toxins or pollutants. And so my research brings all of those concepts together. So to give you an idea of my background, I actually grew up in New York City. So I grew up in Staten Island, New York, which is, most people are familiar with that because it's the ferry that you take to go past the Statue of Liberty to New York City. So I grew up going to the Museum of Natural History, wanting to learn about sea creatures. And I was really interested in scuba diving as a kid. So I went to the University of Tampa for marine science, and I began working with these tiny little creatures in the seagrass beds. So I've worked with the same seahorses and their relative, the pipefish, for pretty much my entire career. So as a undergraduate student, I was really interested in how do we look at where these animals live and how do humans constantly alter the habitats that they are living in. So after I did my bachelor's in marine science, I actually really wanted to understand the biology of these fish and understand their genetics. So I went to Texas A&M. So if there's any Aggies here, gig 'em. And Texas A&M was a great location for me to be able to look at the genetics of these fish and really investigate more about their ecology and evolution. So I went to my PhD, took 7 years. It's a long process of studying these fish. And I got my PhD and then went back to the University of Tampa as a professor for a few years to do some research based out of Tampa. And then after that, I worked with a ton of great undergraduates. So all the stuff I'm going to talk to you guys today about is stuff that either I worked as an undergraduate starting on these projects, and then as a professor got to train my own undergraduate students to do this work. So we've been able to do a ton of this research as a product of having all of these assistants helping. And I've also had other field assistants, such as my dad and some family friends and things like that, in addition to some of these undergraduates at the universities where I work. So this is me and my undergrad advisor who I worked with as an 18-year-old starting out into cygnathed biology, and we still work together to this day. So this is us working together, photoing a seahorse in the Caribbean. So we've actually been working in Tampa Bay for my whole career, but also we started working in the Bahamas. And I want to share some of the research we've been doing in the Bahamas because that is the larger species, the lined seahorse, that we're most commonly seeing, say, at Blue Heron Bridge and along the Atlantic coast. So I work currently at Valdosta State University, and I have a team of students, both graduate students that are master's students and undergraduates, working both in the field, doing field studies where we catch these fish, we photo them, we tattoo them, things like that. We also have studies that we bring the fish back to the lab, and then we do behavioral studies inside the lab with genetic studies as well. So this is my team here at Valdosta, and you can kind of see they give a lot of presentations about their work, which is great. They get to share what we're working on, but we also get to do a lot of hands-on work in the field, catching these seahorses. So you can see in the bottom picture, the bottom left, it shows you a bunch of students with the nets, and that's kind of how we catch some of our seahorses in Tampa Bay. So one of my roles as a researcher for seahorses is a member of the IUCN Seahorse, Pipefish, and Sea Dragon Specialist Group. And so this is a group of scientists that all work across the globe on these unique fish. And we come together several times a year to talk about conservation and how to, protect this species. And so it's a really unique group of people who get to all come together and protect things like seahorses with the knowledge that we've been collecting as scientists in the field. But we also can bring in a lot of different perspectives of people who are doing citizen science and different aspects of collecting information about these seahorses. So something that I would love for everyone to kind of get involved with is I want to introduce my research, but then also talk about how you can get involved potentially, right? So my research focuses on how are disturbances from human interactions altering these organisms, right? And the focus of our research is contaminants in the water, how different things like pharmaceutical drugs and estrogen from birth control pills, how do they get through the sewage treatment facility and then into the coast where all these fish are living? And how do they alter how those fish are behaving and mating, things like that? We're interested in turbidity. So all of the nutrient pollution, like all of fertilizer components, nitrogen and phosphorus, how does that make the water more turbid? How does that increase algae blooms? How does that alter seahorses' behaviors? And then lastly, habitat degradation. We're really interested in when there's habitat loss in seagrass beds or coral reefs, right? How does that change the interactions of these seahorses? And that's really what drives my research that I've been doing for the past few decades. So these are the two seahorses that we work on the most. And the Hippocampus erectus species is one of the predominant ones you see on the Atlantic coast. Hippocampus zostre is the smaller dwarf seahorse. So they're really quite small. You can kind of see the difference between these two. The dwarf seahorse is the adult size is about 3 centimeters. So it's really only about 2 inches long, whereas the larger species is going to be the lined seahorse, the Hippocampus erectus. So most of the research we've been doing has been on these two species. And I'm going to talk to you a little bit about some of the work we're doing in the Bahamas previously on Hippocampus erectus, the lined seahorse. And then Tampa Bay, we're currently working on an ongoing 2 decades worth of projects on monitoring the dwarf seahorse, which is Hippocampus zostre. So these are the 3 species you'd see if you were diving or snorkeling off the coast of Florida. The erectus is the lined seahorse that I'll talk to you about that we study in the Bahamas. The reed-eye is the slender seahorse, so they're a similar size to the lined seahorse, and they're also pretty commonly seen off the coast of Florida on the Atlantic side. The dwarf seahorse are really small. They're a lot harder to kind of find. We often pull nets in the seagrass beds when we're catching them for our research, so it's not something you'd probably see all that often if you were snorkeling. They're really small and really cryptic. So this is something that I really want to share with all of you, because as divers who are out there and finding these different seahorses, getting to be able to report your sightings is an amazing contribution towards some of the research that we are doing here in the universities, because I need to know where the seahorses are, how often people are seeing them. The pictures that you guys take are great because I can often see if they're male or female from the photos. I can tell if they're pregnant even sometimes. So we can really track a lot of information from the data that divers out there are posting on iSeahorse. So the iSeahorse is a part of the iNaturalist program, and it allows people to share their sightings. And in this particular section of the iSeahorse, you can see that there's 40 different species people have reported, and there's over 9,000 observations of seahorses. And the species that I'm going to talk to you today about that is really common on Blue Heron Bridge is actually the northern seahorse, also called the lined seahorse, which is Hippocampus erectus. And this is a seahorse that's found throughout the entire Gulf of Mexico, up the Atlantic coast, from Florida Keys all the way up to, say, New York City. We actually find them as far north as New York, New Jersey. So this species is found all throughout the United States, and they're going to be a much larger species compared to those little dwarf seahorses. So these are pretty commonly ones that are seen on Blue Heron Bridge, for example, and being able to kind of take pictures of these, we can even map identities to some of these fish. If we see the same fish over and over again, we can use a facial recognition program to identify if it's the same fish over time, which is a really neat way to track if the seahorses are staying in one spot, which we tend to know that some of them do that. They tend to have a site fidelity, which means that their location is within a certain territory or a certain distance that they're willing to travel. So in this particular example, they are going to be red listed as vulnerable. And what that means is that part of the IUCN program that the specialist group is working towards is identifying how do we identify if a species is of least concern, meaning that we see them regularly, they're high in abundance, they're not a species we should be worried about, they don't have a ton of habitat loss, or are they a vulnerable species, meaning that they're rarely sighted, their habitats are under threat potentially, and they're not as frequently seen across different habitats, right? Endangered is something that people are more likely used to thinking about, is what is on the endangered species list. But the red listings are really important because we can pull in sightings from citizen science, but we can also pull in scientific publications and bring all of that information together and report on how we should look at these different populations. And currently for the red listing, so this is the red listing for the line seahorse, the Hippocampus erectus. And so each of these iSeahorse profiles have a link. You can look at the link that's directly under the name of the species to see what the red listing status is for this particular species. And for some of these species, we just don't know enough about them yet, which is why any sightings that go on iSeahorse contribute towards our information that we have about these different species. So I want to share a little bit of information with you guys about the Bahamian research that we've done in the past with the larger erectus seahorse. And then I'll talk to you a little bit about our Tampa Bay work and our behaviors in the lab for the dwarfs. So we've been working in a pond down in the Bahamas that is a saltwater pond, which means that salt water is coming in from the Atlantic Ocean into a cavernous location. So this is going to be an alkaline pond, meaning it's salty, and the water's flowing through the limestone in the island's rocks to get into this new location. So there's seahorses that are found in this saltwater pond, and it's constantly concern because if we have any type of farming around the location, any type of contaminants around the location, we want to be able to maintain this population of seahorses because it is such a unique site to study. So what we know about this is that there's limited connection to the ocean and that there's really not a ton of flow from the ocean into this pond that we study in the Bahamas. And there's not a lot of currents or wave actions. The tidal changes are going to be smaller because as the tide rises in the ocean, we see a small change in our saltwater pond where we study. It's not super deep. The max depth is about 30, but it's about a mile long by half a mile wide. So when I say pond, it's actually a pretty large saltwater lake. And so what we have been studying is this population of seahorses. And so if we zoom in on the Bahamas, we're particularly focused on this island here. And so the island of Eleuthera is a unique location that we've been working in for the past few years. And here we can see where we've been studying. And so in this one location, we've actually found seahorses that we've been able to track over several years. And so this is the view from above. You can kind of see that it's a mile long by half a mile wide. And we've been doing research to kind of identify the diversity in this particular location. So we know that there's about 126 different species of fish along the coast of this particular Bahamian island. But in the pond we study, there's only 14 species of fish, one of which is seahorses. And we've been really investigating more about the seahorse biology because it's a location that we can continually survey and find these particular seahorses. So in this particular location, we've done a ton of surveying. The 5 different circles are different locations that we've been locating them in and trying to identify what the seahorses prefer in terms of food that they're eating, habitat that they're staying in. Most of our studies have been done in the shallows, which are right along the edges that you can see kind of the colors here represent the depths that we're surveying in. And so we are able to take the seahorses and take their photo and use that as an information about the individual fish. We handle the fish as small amounts as possible. However, we have gone through lots and lots of permissions to be able to do this research. So I want to add a disclaimer that we do not recommend that that when you're out in the wild as a diver, that unless you're doing scientific diving, it's highly recommended that you don't that you don't pick them up. It will disturb the seahorses, and it will affect kind of some of their behaviors. It might alter the territories that they're staying in. So the work that we're doing here is really trying to investigate how much different interactions are going to alter these seahorses. So some of the research that we're doing is really to better understand the reproductive patterns of these fish. So we need to measure how big they are, how often they are pregnant, things like that we're doing in our particular study. So to investigate their habitats, we're really interested in understanding what type of habitat do they prefer to be in. And we take pictures across all the different locations to see what's on the bottom. And that means that we also then can take a picture of the seahorse, right, with a background, with a grid, so we can measure the sizes and the different proportions of their bodies. So this is an example of a female that we captured on our transect line. And this picture is a bunch of algae with also some shells, which is a typical bottom picture of anywhere in the Caribbean. You have a mix of different types of bivalves, different types of clams, things like that, but also a mix of different algae and vegetation. The seahorses typically like to hide in this, and it's a good location for their cryptic behaviors to blend in. Seahorses can't run away, so they're going to have to just hide. And usually they're going to be very well blended where they are, because otherwise predators can kind of swoop down if there are birds above, or if they're larger fish, they can come and grab them, for example. So my students work really hard to measure all of these different aspects of the fish. So you can see each of the lines or parts of the body that we're measuring to kind of look at differences across adults versus male versus female, to really investigate more about their morphology, more about their body shape. So this is a good example of how to tell the difference of a male and a female. So in the picture on the left, you can see that the one closest to us is going to be a female. She's going to have a larger kind of torso where eggs are going to be found in her ovaries, whereas the male in the back of the picture, he has a pouch that's directly beneath his torso, and so that is going to be a different type of coloring on the pouch structure. And this male is pregnant, which is why his pouch is so large and kind of distended from his body. The picture on the right is showing you a female, and you can see that she has a pretty rounded torso, and that's going to be due to all of the eggs that are currently in her body waiting to be transferred to the male. And the way that this works is that they will have a mating ritual where they'll hold tails, they'll dance together. I have a few videos to show you of what our dwarf seahorses do in the lab when they're interacting. But the female will transfer the eggs to the male, and he will fertilize the eggs as they go into his pouch. And then he will be pregnant in these species. So the species we studied take about 2 weeks. Colder water species often take about 3 weeks of gestation. And then the male will give birth to the live young, all right, after that time period. So when we capture our seahorses for counting them, we usually have a 30-meter transect that we lay out, and we have one person on either side that swims. That way we have each of us serving 1 meter on each side of the transect. We count how many seahorses we see, and then we divide that number of seahorses by the total area that we surveyed, which is going to be the 30-meter transect times the meter on either side that each person is looking in. So that allows us to kind of look at the densities of the seahorses that we're seeing. And we've been able to compare some of our Bahamian samples and some of the Tampa Bay species, and they're quite high in abundance in Florida compared to a lot of these other populations around the globe. So here's some pictures of what seahorses look like when we photo them in the wild. And so you can see in the top left picture is a pregnant male seahorse. So his pouch is kind of the softer looking portion of his lower abdomen. There's a picture in the center with a seahorse that's right next to a brittle sea star leg, actually. And that fish is hiding in the algae. The top right picture is actually the transect with 2 black sponges next to it. And the male seahorse is actually on the smaller black sponge, kind of laying flat next to it. So you can kind of see the pouch of the pregnant male is kind of giving him away on that smaller black sponge. The bottom left picture, you can see there's a bunch of clams that are on in the algae, but we also have a seahorse hiding in the algae as well. And they're really difficult to see until you start to get that visual image. And once you start to get it, you kind of find them a lot easier, but it does take quite a bit of practice. Our students are typically have a steep learning curve, but once they get there, they're really good at spotting them. So these are the two species that I mentioned that you will find off the coast, say at Blue Heron Bridge. The Hippocampus reedi and Hippocampus erectus are the two main species. All of the pictures here, however, are pictures of the Bahamian samples that we've looked at. And so the Bahamian species that we were trying to identify looked really unique because it had a large body, kind of like Hippocampus erectus, which is the line seahorse, but they also had really long snouts like the slender seahorse. And so the pictures really were an important thing to measure. And what we found is that the body structure of the Bahamian seahorses didn't really match either of the samples that we were comparing them to from the Florida erectus or the Florida reedi species that we were studying. So what we had to do to identify what species they were was actually to take DNA samples from our seahorses. So what we do is we actually take a small portion of their fin, and so we are able to snip off a very small section of their dorsal fin, which is the main fin they use for swimming. Now, the reason we do this is because we know that they will regenerate this section of their fin, and that we take a small enough part that it doesn't affect their swimming ability. So we've done tests before where we've taken a small piece, we followed the same individual, we know that it grows back, but they also tend to swim away really well as long as we take a very small portion of the dorsal fin. Now, these are going to be tiny, tiny little fin clips, and then we can extract the DNA, and then we can sequence the DNA. So when it comes to your DNA sequences, every bit of your genetics comes down to 4 letters, right? A, T, G, C's that you can see in the picture here. And those are going to be all of the nucleotides that make up your DNA. We take the sequence of those nucleotides, and the sequence of DNA can be compared across all your different species. So this kind of gives you an example of what we're looking at as geneticists when we're comparing the different species. And so what we found is that we have our 2 potential species, erectus and reedi, and then our Bahamian samples are going to be the ones in the middle on this screenshot. And so we found that our genetic samples matched the species, the line seahorses, the erectus, even though morphologically or, their body structure did not look similar to all the other erectus we had measured from, say, Florida. So what we're interested in is genetically, we know what species they are, even though morphologically their body shape is really unique, which could be a part of them starting to evolve in their own direction as a species when they're found in an isolated pond in the Bahamas, which is pretty cool. So one of the neatest things that we've been able to identify, though, is that the seahorses change their behaviors at night. And so when we're in the water at night, what you'll notice in this picture down below is all of the seahorses congregated in one location. There's actually 1, 2, 3, 4, 5, 6, 7 different seahorses on that one tiny little piece of algae that all clustered at night, whereas they don't tend to do this behavior during the daytime. The other thing is, during the daytime, the seahorses have their heads down and they're laying flat on the bottom, just like you saw with the black sponge where the male seahorse was laying flat next to it, completely vertical, right?
Hey, Emily.
Sorry, horizontal.
Emily, sorry to interrupt. Is anybody having issues hearing us? Please type in the comments section, guys. Emily, it looks like for some reason, when you went to this slide, at least from my perspective, it stopped the feed.
Oh, okay.
So that's why I'm asking people to. No issues. Okay. All good. Okay, good. It's just on my end. My phone. All right. Sorry, guys.
Okay, great. Okay. So what we're seeing here, which is really unique, is this behavior of clustering together. They're also keeping their heads up in the water column, which is really unique. And so what we can see is that their ability to change their behaviors in day versus nighttime is really quite unique. We have not noticed this in other species before. So what we found was that they vary their population densities, how many are packed in one area in day versus night. And then behaviorally, they're not laying flat on the floor at nighttime like they are during the daytime. And then we also looked at the predators to identify how predators around them might be affecting their behaviors. So this is a group of scientists, both my collaborator Tampa and one of our undergrads, who worked really hard with us. And we dove every day at noon and every night at midnight, and had about 4 hours in the water during the day, took a break, filled up tanks, went back at night for 4 hours at midnight, took a little nap, went back the next day during the daytime. And so it was a really, really intense project. So this is the most intense spring break I think this student has probably ever had, because it's the most diving packed into 1 week to survey these fish at the daytime and then go back exactly to the same location 12 hours later at night. So this is what the behaviors look like during the nighttime. Their heads are upright, they're looking around, and when you shine a light, you can see all of the food start to congregate around them once you shine your dive light. So this is a behavior that we've noticed, that they're upright versus during the daytime, they tend to be kind of flat. The other thing that we're noticing is, remember, their densities is how tightly packed they are together. And so as you can see, the daytime bars on the left are really small versus the nighttime. You can see, especially near the shore, we see a ton of congregating fish, especially a bunch of juveniles. And the far shore at nighttime is still also more than twice the density that we see during the daytime. So this is a really cool finding that people have not really looked at, because most of the surveys of these animals are done during the daytime. So if you are going out at night, right, some of the interesting things would be, if you're looking at, say, Blue Heron Bridge, and you go during the daytime, right, you see a bunch of seahorses. How do you go back later and do a night dive to see, are they in the same location? Are they behaving differently, right? Are they interacting differently? And that's something that we're really interested in. So the breakdown of the different groups that we're finding is kind of interesting, because during the daytime, we're noticing that the left bar with the day, half of that bar, more than half the bar, is actually the dark gray, representing the gravid pregnant males. Half of the fish that we're seeing during the daytime are pregnant males, laying there on their side with a black sponge or just hanging out somewhere. But during the nighttime, we notice that in the exact same locations, right, only 12 hours later, females are coming into shore. Either they're coming from the deeper locations, or they could be hiding in the algae, for example. Maybe they're burrowed into the algae and just better at being cryptic and hiding, right? But what we can see is that only 25% of what we see during the nighttime is the pregnant males, and more than that percentage is actually females, right? So we're seeing a difference in the demographics, difference in the representation of the organisms in the daytime versus the nighttime, in addition to that huge increase in fish that we're seeing at night compared to those that we encountered at the same location during the daytime. We also looked at how seahorses' behaviors changes in terms of the body height. So what you can see here is the picture on the bottom is showing them at noon, when their heads are folded over and they're completely laid down flat against the bottom. Whereas when you come back at night, you can see the picture up top is a seahorse holding onto the algae, but its head is upright and they are no longer laying flat on the bottom. So they're changing how they, perch themselves in their habitats while they're holding on with their tails in day versus night. These are also some of the predators that we've seen. We know that octopus and crabs are the two primary predators for these individuals. So these pictures were taken by a photographer we work with in the Bahamas named Shane Gross, and he has been able to take some gorgeous photos of the seahorses. He was able to take the one that I showed a few slides ago with the light shining from behind the seahorses at night with all of the food available. And these are some of the images he captured of the predators that we're surveying as well. And what we're finding is that the predators are actually out a lot more at nighttime than they are during the daytime. So we have not been able to understand yet if the activity levels of the predators is what's causing the activity levels of the seahorses. But what we know is that they are both showing nocturnal behaviors in this particular location in the Bahamas. And we have to investigate this in other locations where we can find dense populations of seahorses, which is actually very, very challenging to find. So We are currently working with the Bahamas government to potentially propose this as a Seahorse National Park to kind of get protection on this location, to have this really unique location in the Bahamas protected. We're also looking at all the genetics behind these fish and seeing how closely related all the different locations in this mile-long pond are. And we're also using facial recognition to look at if we see the same seahorse more than once, because we can actually map their features on their face to identify visually if we've seen them. So this is one of the ways that we do facial recognition. If you look at the bottom right-hand picture, you can see that there's a bunch of red dots on the picture of that seahorse face. And that is a computer program that's been used across a ton of different species, so whales and stingrays, identifying markings and patterns that are repeatable of the same individual. We've also done some tattooing of the individuals, which this is something we have gone through a lot of permission for, to be able to identify unique individuals repeatedly by having unique identification tags. So if you look at the picture on the top left, you can see during the daytime, you can tell a little bit of a pink mark on the back of their head on the top, but you can really see it on the bottom picture, because when we use our black light to fluoresce on the fish, you can actually see that they have a pink tattoo and a green tattoo on their body. And that means that we can identify a unique fish, number 472, say, has the pink on the top, the green on the back, right? And we can tell where we did the particular markings on the individual. We can then look at their facial recognition pictures and compare it with the actual tattoos and be able to compare if we've ever seen that fish before. And so this is something that's been tested in a lot of different fish, salamanders, a bunch of different species, and we make sure that it doesn't affect their survival rates, it doesn't affect their mating abilities, or any of their other day-to-day activities. So this is something that is a really interesting technique that's been used across seahorses globally. We also really want to learn a lot more about what they're eating. And so my collaborator, Dr. Heather Mason at University of Tampa, has been really investigating what the seahorses are eating by looking at daytime versus nighttime to see if there are differences in the type of food that they're eating. And this is something that would be a really neat project for future students to work on once we identify the populations of blue heron bridge that are constantly there, and try and figure out what they're eating compared to what's available in the water column, right? The other thing I wanted to share was some of the current work that we're doing, which is in Tampa Bay. And so we are looking at surveys of fish in the wild. And you can see here are a bunch of my undergraduate students and graduate students standing out in the seagrass beds, where we can take our nets and scoop up these seahorses. Remember, they're only about 2 inches big, so they're burrowed into the algae, and we have to really look real closely for them. And then we can take their photos and then put them directly back into the bay. So this is my student Darcy's project, that she's observing their behaviors in the lab to look at how turbidity and how water clarity affects their ability to mate. We know that there's a lot of different problems in terms of eutrophication across the entire Florida Gulf. So turbidity is one of the main things we're interested in. How does it alter their ability to make mate choice decisions, their behaviors for mating, how well they reproduce, how many babies they have, which is their reproductive success. We also want to look at how survivorship of these offspring turn out after mating in clear or turbid waters. So we can look at the babies to measure their growth rates and look at the sex ratios as they develop, and kind of look at their survivorship over time. So we're really interested in how does eutrophication, right, the turbidity of the water, alter the ability of these seahorses to reproduce. So turbidity is going to be increasing as a result of nutrients like nitrogen and phosphorus that comes from fertilizer. Tampa Bay had a huge releasal of phosphate water from a phosphate mine. And so we know that this can often lead to increases in eutrophication, meaning that the algae is blooming. Most people are really familiar with red tide blooms, for example. And so we're interested in how does the clarity of the water affect the ability of these organisms to interact. So you can see a picture here of 2 different pictures taken in Tampa Bay in a clear environment versus in a turbid environment. And you can see that the visual ability of these fishes might be quite a bit impaired in those turbid environments. So we studied the different dwarf seahorses, and you can see here Darcy's kind of shown us a picture of the male that has the brood pouch with the arrow pointing to his brood pouch. And then the female has a really large, kind of more rotund belly where all the eggs are in her ovary. This male in this picture is not pregnant. So I'll show you some pregnant males in just a minute, and you can kind of see the difference between them. So this is genetically a monogamous species. Majority of seahorses that have been studied have all been reported as monogamous, both socially monogamous as when they interact, and genetically females only give their eggs to one male. The male can only receive eggs from one female. Now they can switch their mates during the breeding season potentially. However, they often are synchronized in terms of the female will give her eggs to the male. And during those 2 weeks while the male is pregnant, the female is generating new eggs and getting ready. And sometimes in the wild, there's been documentation of females checking in on their mates every morning and actually doing a greeting dance together. And they will vibrate together as a way of, while he's pregnant, she's reassociating with him each morning. So the seahorses that we work on have these weird ornamentations. These cirri are the appendages that come off of their body. I've seen some amazing photos from some of the seahorses at Blue Heron Bridge that have these large appendages coming off of them. And that's something that we're really interested in studying, is how these appendages might change over the course of the seahorse's life to help them blend in and serve as a form of camouflage for avoiding predation. So in this particular video, hopefully you guys can see the video. This is a male and a female interacting. And so this particular video shows the orange male with the gray female. And the female is orange because in the belly section, because she actually has eggs and she's getting ready to transfer. And so as they're holding tails and circling, they're also twitching, which is a behavior that we call quivering. And in this particular male, he's not pregnant yet, but you can actually see that he's holding on and showing he's ready to mate. In this next video, let's see. In this one, we're going to have the females holding on to this little fake plant. So we actually use ribbons as our plants in the lab so that they don't block any of our visual of the seahorses' behaviors, and it gives them something to hold on to that they would normally want in the wild. The male is going to be the fish that's circling around her. So she's constantly holding on to the plant. The male is going to be opening his pouch. So you can see that he's kind of bending his body so that his pouch is open. And he's bending it to show her that the opening of his pouch is ready to receive the eggs, which we call male pumping. So this is going to be after they hold tails and they vibrate together for a day or two. Then the male is often ready and he's showing he's ready to receive eggs. That female is really, really large, showing that she has eggs in her ovaries to transfer to him. And then the next part of the process is going to be the heads of the seahorses are going to both point to the sky, if you can notice that. Each of their heads are pointing upwards. His pouch is really, really puffy looking because he's actually filled it with water to puff it large so she can see that he's ready to receive those eggs. He's already pumped and opened his pouch to show that he's indicating he's ready to mate. And now they're pointing their noses up to the sky, back and forth, which is the one of the final stages. In this particular video, it's kind of hard to see, but the female is actually putting eggs into the male's pouch. You can see as she transfers the eggs, the orange increases in color inside the male's pouch. So his pouch is full of orange eggs that she has now transferred. You can see that her torso's gotten smaller because all of the eggs have now been transferred out. So here you can see the orange going into his pouch. It's continually filling. And you can see it gets bigger over time. So hopefully you guys can kind of see the egg transfer happening there. And the female now looks quite a bit smaller than she did before because all of the eggs from her ovaries have been deposited into his pouch. And he's now fertilized them as they go in. And he will now encase those eggs and kind of create these honeycomb structures that hold the eggs in place inside of his pouch. And that will happen over the next 2 weeks. About halfway through at 7 days, there's eye spots on the eggs, and you can see the tails and the eyes begin to develop, and then you can eventually watch the babies be born in the next video. So in this video, you can see a baby is popping out of the dad's pouch and then swimming off on its way. So these are born and ready to go off and start hunting. This smaller species of seahorse that we study is only 2 inches long, and they have somewhere between 10 all the way to 70 babies at one time within his pouch. So he can hold up to 70 babies for 2 weeks. The female will transfer eggs within a matter of days, and then he'll be pregnant for another 2 weeks and give birth to, say, another 70 babies, right? The larger species of seahorses can hold up to 1,000 to 2,000 babies at one time. So this is the dwarf seahorse, which is much, much smaller than majority of all other seahorse species, other than the pygmies that you would see in the Indo-Pacific on corals, the ones that look exactly like the corals. They're really, really small. The dwarf seahorses are 2 inches, whereas the pygmies are about 1 inch, if that. So here you can see we also document some of the aggression. So here is a male and another male, and one male is snapping at the other male to kind of keep it away from the female. So you can see the male that's kind of to the right is getting snapped at because the other male is protecting the female that's all the way to the left. And so you can kind of see these male-male aggression behaviors occur when they're interacting with a breeding pair versus an intruding male. And so there is a level of aggression between the individuals when it comes to pair-bonded individuals that have mated together versus the additional fish that are coming in to gain access. After the male has the babies, we then track all of the growth rates. So in this particular picture, you can see the bottom shows a baby on the day it's born, all the way through over a month of growing. And so you can see the scale is actually in centimeters. And then the inches are going to be the to the right-hand side on this picture of the seahorse here. And so you can kind of see how small they are. They're really quite tiny. If you know that the dad is 2 inches, if you look at the picture with the dad and the babies, you can kind of get a good idea of how small those offspring are and how tiny the babies are. So this is a dad after he gave birth in this picture of the yellow dad versus the dark colored baby. And so there seems to be a really interesting relationship between coloration of the offspring compared to the adults that we're currently investigating. The picture all the way to the top right is one of our offspring that grew up in the lab, and at 100 days old, it actually started developing a brood pouch. And so the male reproductive parts are starting to show up at day 100 for this particular seahorse. They often take more than 3 months, though, to reach sexual maturity. So this gives you an example of a male, what he looks like when he's non-pregnant versus the male on the 3rd day of the study when he received the eggs. You can see the bright orange inside of his pouch. This is a really light colored fish, so the orange shines through really clearly for those bright eggs inside. And so we measure a lot of different aspects of the males, how big their pouches are, how many babies they have, because we're really interested in understanding how to predict recruitment patterns and behavior of these seahorses. So everything that I've talked to you guys about today is due to my awesome undergraduate researchers and my recent master's students that have joined the lab here at VSU. We've gotten funding from University of Tampa, Valdosta State. We've worked with several Bahamian groups. We also have worked with NOAA. And so we have a lot of different groups that have contributed to all of this. And so I just wanted to give them some acknowledgement for that. And so with that, here's another male giving birth. Hopefully you guys can see the video. And so this is another one of our seahorses as he gave birth to several of the babies. And I'm happy to answer any questions that you guys might have for me.
Okay, awesome. All right. What'd you guys think? Wow. It is so cool to see videos of the animals and their behavior. Amazing. Learned a lot, and we definitely have some questions. So let's go into the, you were just talking about the changes of the colors. And somebody asked, they've been told that seahorses will change colors, like just maybe, is there like seasons that they change colors? Does that make sense?
They can change colors in a matter of minutes, actually, sometimes. So when they are stressed out, you can take a picture of a fish in the wild and they'll have these, orange and black spots on them, for example. But if you kind of mess with them at all, for example, if you go to try and pick them up, or if you bump into them, say, they will actually sometimes turn completely black and completely change all of their coloration as a result of stress. We also see this in the males when they're giving birth. They'll change color really fast because they are breathing heavy and their coloration changes as a result of the stress of going into labor. And so the coloration will change when you put them on a red plant. That picture I have of the red with a plant with the red seahorse on it with all the cirri, if you put it into a tank without any type of bright colors, it will often have a more muted color to it. It'll become brown or beige. They'll lose some of those kind of more pretty colors, as you would say, right? And they can change as you move them back and forth between the different tanks even.
Yeah, I mean, over at the Blue Heron Bridge, we photograph a bunch of different colors. I mean, we get white ones, red ones, orange ones, yellow ones, purple. Yep. So, all right. So because we're talking about the Blue Heron Bridge, one of the divers who's a local at the bridge, Susan Gardner, had a question for you about breeding. Is there a chance that there could be hybrids of the two different species, the erectus and the red-eye? Is this possible?
And that's a great question.
Yeah.
So we actually have done, so we had collaborators that were based out of FIU on the Atlantic coast, and they were studying the behaviors of the erectus and the red-eye together. And what they found is that they actually preferred breeding with each other in the lab. And they did produce hybrids that were viable offspring that grew up, that developed normally. But in the wild, we've never found one that genetically shows signs of being a hybrid. And so when we were in the Bahamas and we found these really large male seahorses but they were, kind of more fat in their torso, like an erectus, but long snouted like the red-eye, we were super excited. We thought, oh my gosh, we finally found the first hybrids in the wild. Genetically, we were wrong. But so this is something that we're really interested in looking at. And so because there's very few locations where the two overlap, they're both found all over the Atlantic, but it's very rare that you find them both together and kind of interacting. So that's one of the things that we would love to have pictures of where these guys are located so that if we were to go out and get genetic samples, things like that, we can look into this. And one of the things that we're really interested in is eDNA. And so being able to get DNA from the water, from, say, the poop of the seahorses, we can actually then look at genetically, are we finding these hybrids out there without even seeing the seahorses potentially? So if we know where you guys are seeing them, we can go back to those locations and actually survey if any DNA in the water shows this species, that species, or a hybrid of the two species. Yeah. So we've never seen them in the wild, but that's something we know is a possibility. Yeah.
Okay. Cool. All right. So we talked about the animals at the Blue Heron Bridge. So obviously we've got a lot of people who like to go and photograph them. This question comes up a lot about, well, you talked about not handling them, especially if you're taking a picture of them, you really shouldn't move them so you can get a better picture, right? So you talked about the repercussions of moving them, but what about the lights from the strobes? What is the issues with maybe strobes firing one after another? Is there any issues with that and these kinds of strobes?
So there have been several studies looking at kind of how different interactions with divers affect the seahorses in the wild. And so there are some studies that show that a flash photography in the wild will often it'll startle the fish, but it typically doesn't alter their behaviors very much. They often will kind of freeze in one location. It doesn't usually cause them to go away. When you pick up the seahorse, if you were to touch them, say, that will cause them to move, right? And they're going to move to a different location. It changes their territory. They might be moving away from mates or things like that. And you won't be able to see them in the same place after if you do pick them up. So the problem with flash photography, usually you see a lot of signs in captivity where it says no flash photography, right? And that's one of the problems, is that in captivity, the fish have nowhere to go. They're stuck in that small tank. Some of the fish will actually dart away and actually injure themselves when there's a flash. And so in captivity, it's a really large problem because we do know that that high amount of photography and stress throughout the entire day, right? Or kids knocking on the tank, things like that. That's a high level of stress, a high level of noise in captivity. In the wild, the studies have shown that a single flash often does not alter the fish's behavior. And that's a study that was done in Australia in like 2013, I think, about a decade ago now. And they really indicated that disrupting the fish and moving them was a bigger concern than a single flash. But as we know, in captivity, those flashes add up really quickly and the stress levels go up. And so that can be a huge problem when it comes to the difference between seeing one on the reef, taking a flash picture versus the ones in captivity.
Awesome. And we already know this because you and I talked. Somebody asked, do we have any people doing research at the Blue Heron Bridge? And you and I talked, hopefully in the future, right?
Yeah, that's definitely something that our lab would love to work on. There's not a lot of people serving seahorses in the U.S. actually. So in the state of Florida, we have a few people that work on seahorses, but a lot of us don't have, or we're located at places that only have undergraduates. I'm really fortunate that at VSU, Valdosta State, where I am now, I have master's students. And so this is a project that recruiting a master's student who goes down and dives and say looks at the eDNA, does facial recognition photos, things like that would be a great study to work on. But the more information that we have reported to say iSeahorse at that location, that provides me a lot more information to apply for grant funding and be able to say that this is a location that is a great spot, a hot spot for these fish. Let's go investigate, kind of similar to how we did this in the Bahamas. How do we take all of those techniques and all of what we found there and translate it to now a more local place for us, which is pretty cool.
I think a couple of people, I don't know if you said it in the presentation, or were asking, what is the lifespan of these seahorses?
So in the wild, we're not really sure. We do know that some of the Australian samples that they've been actually monitoring. So they part of this whole tattooing of the seahorses, they know individuals, or they have been able to find facial recognition of individuals. Where the longest one in captivity we know has been resighted over 7 years. And so 7 years for the Australian species is what's been reported in the wild. We know that in Tampa Bay, we captured a small seahorse when he was a juvenile, and we actually kept it in captivity for 7 years. And so that is in one of the erectus, the lined seahorses, which is the one you typically see at Blue Heron, other than the reed-eye. And so 7 years is about what we're seeing both for our data in captivity and the ones reported in the wild so far.
Okay, cool. Well, we're talking about their life cycle. Do they have a pelagic part of their life cycle? Because I've got a couple of photographers who will see them offshore when they're doing their blackwater night dives, where in the bottom zone 700 feet. So they're seeing them in the water column.
Yeah, definitely. So one of the things that we know is that the seahorse babies, or when they're born, they kind of just go out into the wild. They will often hold on to different algae or different types of sargassum, for example, right? And so as that drifts along, they just go for the ride. And so what we know is that both sightings offshore, way offshore, we're kind of seeing these drifting populations, and genetically they are connected really long distances, often because of these individuals who are rafting on these floating mats of algae or sargassum that is really commonly seen, say, off the coast of Florida. And so that is showing us that they are being kind of transported from one location to the next completely by whatever they're floating on. And all of that is supported by the genetics, which is really cool because we can see that rafting behavior occurs in pretty much all seahorses.
Awesome. So we're talking about, different types of seahorses. What other fish is the seahorse related to?
Yeah, so the Signathid family of fishes includes seahorses. It includes pipefish, which are long, straight versions of seahorses. So a lot of the work that I do is actually on pipefish, because the pipefish are really unique in that the males and the females in the species we study look really different. Females are really ornamented, and they actually get to mate with multiple males, whereas each male only receives eggs from one female. So it's a non-monogamous system. The females are actually the ones mating multiply, which is the opposite of almost everything else, which is really neat. So females are the ones competing. They're transferring eggs to multiple males during one single breeding event. And the cool part that I think is neat is that those bands on the female are specific to females when they're sexually mature. And if you expose a male to estrogen, so the estrogen that's synthetically produced for birth control, that estrogen will simulate those bands to develop in the male that is only found typically in females. And so we have the ability to look for contaminants in the water by finding males that have this feminization on them. And so that's something that I've been studying for from my PhD onward. And we're actually going to be studying this summer. We're going to collect fish all across the St. Johns River to look for different locations where we can find these pipefish that are located near contaminants. So pipefish are a really cool system because some are monogamous, some are promiscuous, like the ones we study where females mate multiply. Then you also have pipe horses, which is a seahorse pipefish kind of combo. Pipe horses are predominantly found in Indonesia area. We also have sea dragons as the kind of last part of that group. And the leafy sea dragon is the one you see in captivity. The weedy sea dragons are also pretty common in captivity. Wheaties we've been able to mate in captivity, whereas the leafy sea dragon, we've not been able to successfully mate in captivity. So one leafy sea dragon is brought in from the wild a year, according to the Australian government, right, which is the only place you can find them. And then those are the only offspring then that are available to aquariums and, educational facilities like that. So that's why it's really hard to see them. And that's why they really ask you not to do flash photography, because those individual fish are, going to be pricey individual to be able to have on display. And they often have a lot of health problems because they're just not, they're not as well prepared to be in captivity compared to those captive bred seahorses that people have been breeding in captivity for a really, really long time. So we've really mastered some of the breeding of seahorses for aquaculture so that we don't have to remove them from the wild.
Absolutely. And that actually brings up, we're talking about breeding again. Another question that came in is, are sounds from the reef a factor in seahorses dropping off the sargassum and settling into the bottom? I've got a diver here saying that they've watched a few drop and they seem to be able to hear the clicks and sounds from active part of the substrate.
Yeah, so the seahorses, typically the reason they're rafting is because they're just not very good swimmers, and once they latch their tail on, they kind of just go wherever that goes. Often you'll hear the seahorses, though, snapping their jaws as they're eating. When they snap their mouth forward, they're creating a suction that's really strong. And seahorses are one of the most accurate feeders of fishes. So they've done a lot of feeding studies across a ton of different fish, and they found that the seahorses are super accurate in achieving, their prey items. And seahorses don't have any teeth. They don't have a stomach. So they are forced to kind of suction feed everything as it goes by, and they snap their jaw out, and you can hear that noise as they're feeding. But since they don't have a stomach, they don't have ability to store food. So this is why you have to feed them so often if you have them as a pet. And then the ones in the wild are continuously eating. So our feeding studies have shown that when you look at what's inside the stomach, you can kind of flush out to see what the seahorses are eating, right, in some of our studies. And their stomachs are relatively full throughout the entire day and the night because they're constantly just consuming things around them because they have no stomach to store it in.
Awesome. And a question that came in about predation. You talked about crabs and the octopus as their main predators. Is the lionfish, now that it's been introduced to this area, is that an animal that will eat these guys?
Yeah, so so lionfish have a huge gaping mouth, so they can kind of eat anything and everything. And so the seahorses have no ability to get away from lionfish. And so the likelihood of them being inside a lionfish is pretty high. And so there's a lot of other fish in the actual ocean that we see that are eating them. There's pictures of mahi-mahi, for example, with big stomachs full of them, you can see tons of seahorses inside. But in our particular location, there's not a lot of fish diversity. So we're really not seeing a ton of predators there, which is why the octopus and the crabs are the only ones we've seen eating the seahorses. But in the actual ocean, the rest of the locations where seahorses are found, they have a ton of other predators, right? So there's cool pictures of people documenting birds that have grabbed a seahorse from the shallows and flying away with it. And there's a ton of different species. Pretty much, I think they're just a good snack for almost anybody, it seems.
Yeah, and I know that there's some other divers on here that are listening. I mean, I've been at the dock where the boat is, and you'll have in the sargassum, they're just sitting right there, and it's like, wow, you can be snatched up real quickly because you're just sitting right there on the surface. So anytime you guys see floating sargassum seaweed, check it, because you're gonna probably find a seahorse in it. So somebody asked, what is the season for seahorses at the Blue Heron Bridge? I'm not sure you know this, but guys, right now, go dive the Blue Heron Bridge. It is full of seahorses right now because they are all mating with each other. If you didn't hear the first part of my presentation, we are seeing photos coming in of pairs mating, and I think somebody that Brenda Hill was diving with, they got the male pumping out the babies. So this is the season. I guess they like the cold. It's about 75-degree water temperature, and they are there now. So, but usually, I mean, there's year round they can be there, but there's more of a majority during this period at this point right now. So if you guys want to see them, this is when you go. And if you see them, like Emily said, go to the seahorse.org and you can register your sighting with your photos. I know that they would really appreciate the feedback from us divers. We are the eyes and the ears of the ocean. We're in it more than a lot of these researchers. Sometimes they're in labs and they're behind computers. So if you guys want to help out with seahorse research, go to the seahorse.org website. And guys, it is time for our raffle. So I'm going to go ahead. I'm going to hide the presentation that you have, so that way I can bring this in. Okay. Oh, here it comes. Boop. There we go. There it is. All right. So, guys, if you want to go and see seahorses other than the Blue Heron Bridge, we've got a lot of travel opportunities. I talked about this in the beginning. So there's that travel banner. Just click on it. It'll take you to all the cool places that we're going around the world. Maybe you can go see one of those little pink pygmy seahorses over in the Philippines with us, or Raja and Pot. So go to our website, and then if you want to go and do a dive with one of our dive guides, we're in the water almost every day at the Blue Heron Bridge, so we know where they're at. You can always hire a dive guide from 4C. You can go as a group, or if it's just you, you can pay for the dive guide to take you out, and they're going to know where all the cool critters are hanging out. But we are going to raffle off a dive guide with one of our famous Blue Heron Bridge guides. So I'm going to go ahead and pull up the random name picker. There's everyone's names. They're all been transferred in. So let's see who the lucky winner is. And James Reap. James, if you are watching, please give us a shout in the comment section going, woohoo! All right. So James, we're going to get you out there and we're going to get you go diving, see some seahorses. Make sure you wear your wetsuit because it is a nice 75-degree water temperature. And congratulations on that win. So I'm going to go ahead and wrap this up, guys. If you have more questions for Emily, you can either email us at info@force-e.com, or Emily has a couple of different ways to get a hold of her. Go ahead and ask her more questions after this, and we appreciate everybody tuning in. Dr. Emily Rose, thank you so much for a fantastic presentation. We appreciate it so much. Looking forward to working with you in the future, hopefully getting you guys to come to Blue Heron Bridge and maybe doing some presentations about other types of animals that you're working with. So, all right. Thank you, everybody. Have a great evening. See ya.
Thanks so much.